you so much for the nice opportunity to speak here and tell something about our work. So I'm Roeland Merks. I'm a biologist by training. I'm now at the Mathematical Institute and the Institute of Biology at Leiden University. Also, as of September 1st, I will be taking on an additional challenge because I will be director of the Roeland Center, which is a workshop center very similar to the Newton Institute, but covering all sciences, including the humanities and social sciences. So, so what I'm going to talk about today is cell-based modeling. And if my laptop wants to cooperate, there we go. So in my group, we work on it's called multicellular mathematical biology. We work on a range of topics of angiogenesis. So I'm showing here a couple of examples. I'm showing here on models of angiogenesis. We're currently also doing experimental work to validate these models. We're also working on copying of the pacemaker or the sinusoidal node cells in the R2 atrial cells in cell culture, and trying to understand what should be the properties of the boundaries between those cells to get proper signal transduction and no exit block. We're also working on cell migration patterns. I'm going to tell you some about that today. But then also we've worked on mechanobiology, some multiplication due to strain, for example, and also on taxes. There's also this week paper came out in biophysical on this work. I don't have time to talk about this today. So basically what we're doing in this work is what we call cell-based modeling. And in cell-based modeling, what we do is to start with some description of cell behavior as input. And what we hope to get out of that is the development of multicellular structures. So the growth and form of tissue. And the aim of that is to understand how cells build tissues collectively, but also how tissue structure feeds back onto cell behavior. And of course, also if there are modifications in cell behavior, how this convention works has an effect on what happens in the whole embryo. And in order to do that, in order to really go to the cell and multi-cell scale, what we need to do is to put in sufficient detail in our single cell models. So, so as to capture the emergence of collective behavior, but also we should have computationally efficient models in order to simulate lots of cells together. And this is an example of that, where we have captured single cell behavior, in this case, an elongated cell. This cell secretes some kind of chemofractant and attracts other cells. And then out comes the collective behavior. Well, if this is now the right way to describe angiogenesis, that's what we're going to get in the lab, because we also have a dozen other ways to model collective cell behavior. And I'm going to model network information. I'm going to show you one other example of that today. So there are lots of ways, as I think many of you are aware, many different ways of modeling single cell behavior. And of course, some of those methods use single particles on the lattice or off the lattice, and sometimes have a small volume or some shape. But in many cases, it's really important that the cell has indeed a particular shape. It can do something else on one end, and it can do it at the other end. It can polarize, or it can be contact inhibited by adjacent cells. So what we typically do is stay on this side of the spectrum, where we use multiple particles to model cell behavior. Well, and then one particular method that we really like, and that I know that some of you also like a lot, is the cellular pods model. And I'll, for those who don't know it, I'll briefly go over. And basically, what we do in the cellular pods model is that we have project our cells on the lattice. This can be two-dimensional, like in this example, three-dimensional. We project it, and then we have multiple lattice sites that describe one cell. So, for example, here I have three different cells. And I have an environment in which those cells are. I will tell more about that environment later. But for now, we have three cells in some empty environment. And what we now do is that we say, well, all these cells are at exerted forces on one another. For example, they may want to contract. They may want to pull other cells. They may want to move to chemoreactant, something like that. We put all those forces together in a Hamiltonian which is also a way of describing this force balance. And why do we use Hamiltonian? Just because of convenience, because of computation. So what we have here is the classical cellular force model was introduced by François Damier and James Glacier over 30 years ago in 1992. By the way, we are also going to celebrate that by the end of this year at the Roeland Center with a workshop on the cellular force model. So I hope to see many of you there as well. And the two pioneers, and also in hopefully we introduced hybrid cellular force models will also be there. So what we will do is to we have this classical cellular force model. There are two components to a Hamiltonian, and one is the cell adhesion, and we have some kind of contact energy between the cells, which is the adhesion energy, and we have a volume space where the cell wants to move to a particular resting mode. And what we then do, we use the Prof dynamics to update this system. It's very similar to this spin model. We pick a random site, then we pick a random neighbor. We consider an energy change, delta H is the acceptance copying. And if this reducing energy apparently was force acting in this direction, and if not, we accept it with Roeland Merks to mimic also active utility of cells, the energy that cells are putting into moving randomly. And what I forgot to tell you is that indeed we represent ourselves by numbers, of course, in these letters, where we have ones, for example, there, and twos and threes, and these are the spins, and T tau is some kind of cell type.
Can
Yeah, sure.
So if you have a single cell, what happens? It's good, you have no interaction there.
So in this case, if we have a single cell, it will go to its resting volume, but also it will actually go a little bit smaller than the resting volume, because it also tries to minimize the adhesion end or the surface tension. So it will typically go a little bit beyond smaller than the resting area, such that these two energies are balanced out. And we can add all kinds of additional constraints. So I told you that in this example, typically this is used for cell sorting, for example. It was introduced for modeling cells. I can tell you offline much more about that, because what I want to go on to now is extensions of this framework, where we are going to put extra detail in this empty space, the medium that I am more or less ignored. Because we know that in this empty space, there's a material in many tissues, the material of the extracellular matrix. Extracellular matrix is a network of materials, collagens, carbon, nectin, elastin, what have you, all kinds of materials that are in the cellular environment. They are ways to glue cells together, but they also provide means for signaling. And what's special about this kind of signaling over traditional kind of molecular diffusive signal is that this signal can be long-lived and long-distance. Why long-lived? Well, because the cell can modify the extracellular matrix, it drop a signaling molecule there and walk away, and then another cell will pick it up and respond to it. And why long-distance? Well, because it can pull a fiber, and then the cell somewhere else can respond to that force in the fiber. So these signals are long-lived and can be long-lived and can be long-distance. So we really have to consider the ECM if we really want to understand tissue morphogenesis. Then all of the examples that I showed in my introductory slide, they had only minimal model of these things. And so what I'm going to do now is show you how you can introduce the ECM into the cellular force model. I'm going to show you two ways, or three ways actually. First, I'm going to show how you just have some kind of piece of substrate, how actually can cell motility be modified by this substrate. Then I'm going to show you an example of explaining how stiffness of the substrate can change cell shape. And then I'm going to show you a very recent development where we realized that the extracellular matrix is very structured and has lots of fibers. So I'm going to show you hybrid cellular plots in molecular dynamics method to model the extracellular matrix.
So the case where there is stiffness is extremely rigid, the object is the case without ECM, no? So the previous results are
Yeah.
related or not.
Well, that depends. You can see there's an undescribed empty space because it could be a very stiff or a very soft surface, depending on how you interpret it. But in general, you could say.
No, but in terms of the model, so your previous model it's very stiff
Well,
or not.
I'm going to show you some examples where this may be different. That's why I'm kind of hesitant to say that it should be very stiff. It really depends on how you interpret the model, I would say. So this is a bit of a messy slide, but basically, what I'm going to show you now is to introduce hybrid cellular. So what we now have is our cellular plots model, and we couple to that some kind of other model to describe the extracellular matrix. And these can represent intracellular fields or extracellular fields, things like diffusing chemicals, some kind of electrical potential. I showed you some examples that in the introductory slide, but also stiffness, strains, etc. And these can then be all kinds of things, whichever you like. We use PDEs, molecular dynamics, some other particles. But the essence of that is there's a mutual coupling between them. So the cellular plots model is going to respond to the field. For example, if there's some PDE describing a chemical gradient, then the cellular plots cell might respond to that by moving preferentially in that direction. But also, the PDE should be dependent on the cellular plots model. So, for example, we have some extra source terms at the place where you have a cell in the cellular plots model. So there's really mutual interaction between them. And an example is indeed, suppose we now have a PDE, something it's a very simple PDE, something like this. Term, degradation term, and maybe some source term where we have a cell. That's usually where this identifier of the cell larger than 0. Then we could say this will then generate exponential gradients around the cells in equilibrium, and then in steady state. And then what we can do is that we add an extra term to this Hamiltonian, depending on the gradient, local gradient of this chemoattractant. And that is very important that we can really easily change between forces and energies just by calculating if the cell is trying to extend, trying to make a copy, that we multiply the force that it wants to exert with the distance, and we have simply our energies, our work, and this we can then put in a Hamiltonian. And in this way, we now have an active matter model. Okay. So I realize that I put things a little bit in wrong order. I will briefly show you then an example where we do exactly this. And that's precisely this model. So this is where we do it. So here, every cell is a source of the chemoattractant that secretes and diffuses, and that the cells move up to the chemoattractant gradient. Usually, we can lead to clumps of cells, but since the cells are elongated, they form that. I can tell you the details of all of that, but I want to really focus on the extracellular matrix. But it's a kind of an easy example to show hybrid cellular. So let me start off by having a really simple description of the extracellular matrix, and that we introduce to try and understand how the ECM can understand migration patterns of cells. So this is an example by our collaborators, Universiteit, and this was also done in the University of Toulouse, where they found that a B cell line where shows different migration patterns depending on the substrate we are on. So here you see the cells, and it turns out that on collagen form, the cells form only very small adhesive patches, and a fibronectin they form larger adhesive patches. This is the third microscopy. So you see where they really attach to the substrate. And indeed, what you can see is that on the collagen or they have very small migration, very local migration, and the fibronectin they have much more wide migration.
So, sorry, what are these? This yellow line, I mean, this.
That's the cell in the fluorescent microscopy.
Oh, I see. Okay. Okay.
And I have to say we didn't fully crack this problem yet, but we could at least have a model where we find where we can explain differences in migration. But basically, so basically what you see is the full cell shape and where the cell really adheres to the cell. So how can difference like this occur in migrating cells? And our hypothesis was this is due to cell substrate. So what I'm showing here is a model, an extension of the cell model, called the ACT model. Again, this is a thing I can explain in detail later on. But basically, what it says is that on top of the cell plots, you have an activity level that tells you how recent did the cell copy. And if then a cell recently copied, it has to compete with another side of the cell that didn't copy the one that recently copied with. So this way you can kind of introduce some kind of persistent motion. And it's a kind of force-based model of actin polymerization. That's why it's called ACT for activity level and also actin. And basically this was introduced by Universiteit over. And basically in this way you can create really persistent cell motion. Now what we did is introduce cell substrate adhesion to this. So now we say that at random in the cell, an adhesion can occur. You can have the adhesion can grow according to the even growth type mechanism. If the cell then tries to retract, and it has to, there is an adhesion on that. It has to pull away this adhesion. Then we introduce an extra energy. It costs some work to do that, but it reduces the retractions and also they can disappear. And what we now see is that if we, so this is, we have very slow growth of adhesions, we get kind of typically the behavior of this ACT model, which based quite nicely with several laws or several observations on cell migration, including the.
So what are the darker green points?
Yeah, exactly. Those are the adhesions.
The adhesions, okay.
Yeah.
And the red one, so the color coding.
So those is the high activity level. So you can see this as where the place where active polymerization takes place, but it's strictly it's active. Yeah.
And the adhesion side is always. What do you mean by
Yeah.
growth rate?
Sorry.
How do you implement the growth rate?
There are two ways we can implement those. One of those is by random appearance of adhesions, and the other one is by having an even growth mechanism where we have a high probability next to an existing adhesion. So as a certain adhesion kind of grows as a patch. And what we then see?
Can I just ask computationally, how heavy is this?
The super light. It's actually so light you can play it on your iPhone. I show at the this is a nice work with cellular package developed by Immortal in Nijmegen. So when she saw a preprint, she immediately started implementing this, and I said, "Oh, this is so great! I want this to be in the paper." So in the revisions, she joined us, and so the model is in the supplementary material, runs in any web browser. I will show the QR code in a minute.
So you don't have a preferential size, depending on the size or more the form of the cell, where to create these
Sorry.
compositions.
Yeah.
Where do you create? Just randomly.
Okay, let me. Yeah. There's two ways. One of those is sudden disappearance, and this can be dependent on the activity level or different parameter. And it can be due to we have a higher probability next to existing adhesion sites specifically. once the cell kind of rolls on, it kind of rolls out and forms additional lesions next to it.
And the energy substrate, energy to from these focal adhesions is just that the energy is it's more favorable to them to stay there.
Yeah, yes, yeah.
And
that's-
the other parts.
That's also the main effect of the cellular fox model, the dynamics of the cellular fox model is at the axis.
Yeah.
Because that's where when the cell tries to infect, if there's too many adhesions there, it has to and that introduces a lot of energy, so it has to make many random movements in order to pull away. So and then there was also some variance of the model. We also looked at having the feedback between the activity of and the adhesions of the and the appearance of the adhesions. And so I'm kind of really want to go very briefly over this this work because I have so many other things to tell. But let me see. Yeah, but basically what we can see here. So this is kind of normal persistent motion. You have it's really diffusive. And if we introduce more and more of these adhesions, it goes to subdiffusion. And when we started this, when we showed it in the paper, when we started the work, I really thought that we could also use this model to get more super diffuse behavior. Because once the cell is kind of moved loose, it kind of takes off and it has to kind of stop before forming new adhesions. We didn't get there yet, but yeah, I actually think we should study some more parameters that we didn't study them. Because in this paper, we mostly studied the TS parameters, so the random appearance, and not the X-pro. And I think actually, if we look more into that, I still have to help it. So it's kind of not completely finished to my feeling, but there's actually already a lot of nice results in this.
You can define polarization in this cell, right?
Yeah. So the way the polarization is on here is using this this ACMO. Maybe I should explain the ACMO offline because
Okay.
there's actually lots of it's a super elegant way of describing cell polarization. So basically we vary these parameters and we can get this kind of subdiffusive behavior that we see here in B and D mostly, and also kind of stop motion there. So this is really the idea. They move, then they start forming adhesions. They can stop once you manage to get sufficient adhesions. It slows down the cell, but allows them to make more adhesions. But then, if a new polarization form is formed, it kind of takes off again and starts breaking up pieces. And then, so this kind of creates this kind of stop motion. Anyway, just looking for different things. But so what we see here in red is the adhesion area of the time. And you can see that for some parameters you find some oscillations, and also for some parameters you see really this large variability in cell speed.
So, Roland, let me ask you. So, on the experimental data, so on the experimental data, you look at the velocity correlation option.
Yeah.
What do you see?
So the ACT model without
No model, just on the data, on the cell. You track, let's say, the symptom, you look at the velocity correlation option.
So we didn't check this for these immune cells. So basically what we show here, well, first of all, the ACT model without any adhesions. It's basis law where the velocity is correlated with with persistence. So I forgot the name of the model, but there's a really nice model that's been shown in the paper by Numerical, where it shows that this behavior correlates with many cell behaviors.
But you have an exponential decay, or it becomes negative with oscillation that tap out. So just that gives it's a decay. So the persistent typically is
Yeah, we mostly characterize this by looking at the mean square displacement. And so there we see that the typical behavior of this is quadratic at first, when you have your persistence time, and then slowly you start having turns in CPMs, and then you go to this typical diffusive behavior. And but for subdiffusive models, and of course, then at the longer term, you have instead of the square behavior, you have a lower exponent.
Because in general, when you look at the plot, the correlation function given is from the model. It's better than looking at the MSP, it's more precise. In fact, it remains positive, and that's your persistence.
So I say it again.
It's better. I mean, on data, it's always better to plot the velocity correlation function.
Yeah.
Because if you have a velocity correlation function, it's always positive and it decays, it's exponential. That's a typical persistent case.
Yeah.
So long times it's diffusive, but short times your persistence there, and you can even measure.
Okay. Well, that's
and then instead of having velocity correlation function that oscillates with damped oscillation, but it's also negative. That's the case that gives you some sort of fluid.
okay. We'll have a look at that. Thank you. Thanks. Yeah.
The velocity correlation function is the second derivative of that.
Yeah, but on the data, typically you're right, but then typically the data is always better to look at the velocity correlation function.
Well, it depends, because you have some, at least in our simulations and in some experiments we have analyzed. What you see that if you go to very small short times, and what short times means depends on the cell and your cell. Okay? But if you go to very small, it's not ballistic anymore, and it will be very short time diffusive. And in that time, you cannot define velocity.
Okay, well, I mean,
So
yeah, okay, but in as much as you have two temporal value, you can still define that correlation. No?
yeah, but
So,
what are you going to say if you are in the diffusive? With short time diffusive, what are you going to say?
I'd love to discuss it offline, because I think there are many more.
let's discuss later.
I kind of wanted to give a glimpse of how it works, and I left out all the details because we looked at so many different ways of describing this data. I don't think we looked at the velocity of correlation function. We should have, but we didn't. But I'd love to discuss this. But I just want to show that indeed we can use the model now to describe a lot of different cell behaviors, but not that super diffusive behavior, which I really hope we could do. But already a lot of behaviors. I'm really happy about that. Because what I want to go to next is how we can now use hybrid cellular plots model to understand the effect of stiffness on on cell shape. And well, this is just a slide to show that many cells, fibroblasts and epithelial cells, what have you, cardiomyocytes, if you plate them on a substrate for different stiffness, they change their size and they change their shape. So typical behavior that is looked at is spreading behavior, but also typical behavior is elongation. And the common way of looking at that is, I would say, a mechanical crossflow. So where the cells are trying to contract, then as a result, the substrate strain stiffens, and this has an effect on these little adhesions that the cell used to hold onto. And in the previous model, we just had very generic adhesions, but now we'll make them mechanosensitive. And as a result of a higher tension for adhesions, they tend to mature, they tend to become stronger, so they can hold them more strongly to the substrate. So now let's see if we put all that, what happens if we put all that in a cellular plots model. And I'm going to start with a very simple one. And that's why I said, well, it's so hard to say whether the substrate is considered soft or stiff. Because here we still have a very generic description of the environment. What I'm simply saying is that I now have these focal features that I put in the initial model. I now make them a little bit more dynamic. So I make every focal feature I make a little, put a little ordinary differential equation there. That will say where that will grow depending on the force that it feels from the cell. So basically what we do, we have a kind of three-way hybrid model. So we have a cell-pulse model. Now I'm predicting traction forces within the cell. I'm using for that first moment area model that was used by Roeland. I'm then, so basically it says the further you are away from the center of mass, the larger forces. Then I'm giving these forces to ODE models. And these ODE models say that if the force is larger, then so these are the models basically. So we have the size of the propagation. So there's a growth and a degradation. And we said that the degradation is going to be force effect. So if there's tension in this propagation, the degradation is going to be suppressed. So then we assume that tension in the propagation is building up over time. So this is due to the model by Sparks. And basically we say that if you're on a soft matrix and the cell starts pulling, it takes a little while before it's in the chemical equilibrium. But if you're on a stiff matrix and the cell starts pulling, it really reaches the stalling force really quickly. And so then if you look at the integral over the over this short time into equilibrium, then of course on the soft substrate, the focal lesions have experienced less tension than on the stiff substrate. So that means that on the stiff substrate, the degradation is suppressed more than on the soft substrate. So that's basically the story. So that's basically the story to start. So what did I put in our model is to say, well, first of all, I'm going to change my Hamiltonian. I'm going to say that the cell wants to shrink completely to zero size. It still has to observe extension, but it also wants to spread out into the generic region. So that's a must. So then I say that whenever the cell wants to retract, very similar to the previous model, if there's a population, some conditional energy is applied. And this energy is now not the same for every adhesion, but now it's going to be dependent on the size of the population. And that's basically what I say in this. So a bigger population takes more energy to pull away than a smaller population. So let's see what happens. Whoopsie. What happens if we put that in a model? So here I have the behavior of the model. You see that these populations are these black dots. So the cell slowly grows. And whenever it kind of gets a new location, a new population is formed, and it starts pulling on it, and if there's more force, and if the substrate is stiffer than the population will grow, and it will take more energy to pull it apart. So you get some kind of ratchet effect that makes the cell spread. And of course, then on soft substrates, the population will simply degrade. So it just spreads a little bit, and the stiff substrates now still spread out. And the result now is that we get cell spreading. So that means that we have now understood cell spreading, but not yet cell elongation, which I found the most exciting thing to try and explain. So what we have to do in order to explain that is now we have to put in more detail of the substrate itself. And we're going to introduce a finite element model to model the deformation of the ecosystem. So basically what I do, I have the same model. So I have my cell-pulse model, my traction forces. I feed those traction forces simply to a linearly nested isotropic finite element model, and I get from that tensile stress. And now the tensile stress is going to affect the growth of the lesions. And we have to say more. So the way I do that is by saying that if there's planar stress, this leads to additional recruitment of stabilizing proteins, balance vinculants, what have you, and they will lead to extra growth of these local pieces.
So you are supposing the substrate is homogeneous? No?
At this moment, I'm assuming the substrate is isotropic and linear investing.
Yeah.
but but it's not homogeneous because it can, the strains can differ depending on the input. And the next step, I'm going to take also away the isotropy in the linear elastic part. But first this. So what do we get? So I have here again my soft substrate. So the cells just try to make adhesions and not much happens and they just slip off. So now the substrate stiffens. And well, once they make a cohesion, they also start deforming the matrix. Now I'm making it even more stiffer. And what you now see is that slowly the cell kind of polarizes, or this is really polarized, it becomes elongated. And you see two locations on either end where, of course, now eventually you have to large spores. But also what helps is that once a competition starts growing, it can form. But it can also help the additional competitions to grow as well, because it generates pain and stress. And so the additional competitions can grow as well. So slowly, so elongates over time. And so what we see now is that we look at the stiffness of the matrix, you look at the cell eccentricity, that on soft matrices, the cells still stay rounded. Also on stiff matrices, they still stay rounded because every time it makes a competition, it just sticks directly. And at the intermediate places, the cell elongates.
What limits the process? Those conserved volumes at all, or?
There is no conserved volume in this one because we cell tries to go to zero, but at the same time it tries to spread.
But you could stretch to infinity, or
no, that's what you mean. No, it could not stretch to infinity because then the substantial starts setting in, so that reduces the cell shape. And also there's this force going back to the zero area. So if you don't have any interaction with the finite element model, it will simply spread a little bit and stop. Because then it's a balance between the spreading part of the homogeneous, the negative component, and the component dragging back to zero, and then you have possibility to stay. And indeed, also, if we look at the dynamics of cell spreading, this was this little simulation I did before preparing another presentation. We didn't tune it quantitatively, but what I like to show here is that the way it starts elongating is a sigmoid way, and this is also what we observe in spreading cells. And this actually thing how to put to show to a student so that they can really figure this out in detail. This is just a little thing I wanted to show them. Of course, we can tune all kinds of parameters. In this case, we have the velocity by which the cells are pulling. So if they pull really fast, they reach equilibrium really fast. So then already in soft substrates, competitions start maturing. But if we let them move back really slow, you see that the elongation behavior only occurs in very stiff matrices. So this could be one aspect that could also explain cell type differences. And so what I really like if such a model also can explain, apart from cell elongation, it can also explain other phenomena. And for example, what we have here is the cell in the stiffness gradient. And let's see what happens. Because now the cell starts elongating, but also since one end of the cell is now going to be on a different stiffness than the other end of the cell, then on the stiffer side, the focal adhesions are going to be a little bit more stable. But then over time, the cell slowly drifts, or I have to go this way, slowly drifts towards the stiffer side, which is a phenomenon known as neurotexis. And of course, we can measure all kinds of things of that. We can cell trajectories and then look at cell speed. And cell speed indeed depends on the slope of the stiffness gradient. How much time do I still have? I'm
20. Right.
oh, so okay. I'm happy to discuss any detail later on because I really want you to see this this part of the work where we take away the isotropy and well make it heterogeneous. So because it was of course not correct to model the ECM as an isotropic material, so now that's why we introduced a new method. And that will just publish this this week, and postdoc did this work together with another postdoc. She made a really nice visualization that made it to the cover. So I'm really happy with that as well. And what you see here is indeed, I will show you the details. Basically, we have a contractile cell. It tries to go to 0 again. We have focal lesions there. They contract, and then we have an actual matrix that. So now many people ask me, why did you do this in a hybrid cellular plots and molecular dynamics model? Why not simply make this, for example, using the subcellular animal model, also put this into molecular dynamics? Well, the reason is that with this this method, we can now also use all these other methods that we have developed for the cellular plots model and really combine those to really start solving interesting biological problems. So it allows us to kind of take away all these other innovations we already have for cellular plots. So what did we do? So basically, here's again our cellular plots model. I now have for the time being only one cell, and here's the molecular dynamics model. So they're simply overlaid as we do typically in hybrid models. So basically, we say we have now these focal lesions. And now these focal lesions are associated to a particular node in this molecular dynamics model. The fibers are modeled as bead springs systems and with angular springs. So now every time the cell, and this is simply done using typical over-down dynamics plus some random noise. So now we simply have our cellular plots model where the cell might retract a little bit. And once this side that is retracting is associated with cellular plots, so with the bead of the fiber, the fiber also goes along. But now, of course, we have a problem because this fiber will be attached to all kinds of other stuff. And so now we have to do these kind of things that you typically do in these computational models, that we kind of do, I say it, an operator splitting kind of approach. So we, once the cellular plots model is retracted, we kind of freeze the whole network. Then we only look at the local springs. And those local springs, we can simply calculate those for simple springs, and we turn those into energies by multiplying the distance over which the cells retract. And so that we can again put, this is one of these work terms that we can put back in the cellular plots model. So, oh, the equation is not there, but it's simple spring equations that we put in. So once that is done, so then once several of these sides have been retracted, then we go back to a couple of steps in the molecular dynamics simulation. Sorry? Yeah, exactly. Yeah, indeed. So that's good, really good point. Yeah. So the molecular dynamics is off the grid, and so the plots model is on the grid, and this creates also some bookkeeping problems, and those really took the major part of the development to do this efficiently, and we're still working on making it even more efficient.
So the covers are all levels.
Yeah.
It's kept as a different field.
Yeah, it's really even a different software package. So just to give you a little bit insight in the very rough insight in the technique. But basically we use the standard open source package for molecular dynamics, blue. Then we have our in-house cellular code, which is also open source. We also have more recent versions which happy to share with you. And this whole code is actually also on Zenodo. And then Hill wrote a PyTXT wrapper. So she wrote a wrapper around our cellular plots code that allows us to couple this routing with the cellular plots code. And so this is now the continuous development. So we now have assistance from an engineer from the Netherlands eScience Center, who's really helping us to replace this with code coupling things. There are things like muscle. There's also things that we use, which are typical ways of coupling these different codes and doing it really efficient and very standardized and reusable. So that's just a little glimpse into the really hard work that is under the hood before making this possible. So I'm showing you some examples. So here you see a very soft matrix with very few cross-linkers. Yeah, please. Sure. Yeah, that's a good point. To be honest, we basically did a trial and error at the moment. But one of my PhD students now started thinking quite deeply about it to see if we can really say a little bit more formally about it, because it's an important form in general also in these kind of hybrid simulations.
Yeah, no, for the athlete, we're going to be able to do it all at the same time.
Yeah, and yeah, exactly. And so there's different, so there's actually lots of different aspects in there, because there's of course on the one hand you want to suppose you simply have do one thing and then the other thing, and then so then you want to minimize the amount of these steps before going to the to the cellular optimal, and this we can figure out by trial and error. But also in terms of software engineering, it's often very much more interesting to have the molecular dynamics model running continuously on something like a GPU, and then regularly probe the from the cellular plots model. So these are the kind of things we're working also on with the engineer to optimize these kind of things. But at this moment, what we're doing is indeed have a fixed number of steps in the Hundi simulation. Then we have one non-target step where a number of these subtractions are made. These are then communicated back. And so if you let it run, we're now just at the moment where we could probably reduce the amount of Hundi time and still get the same results, but we're kind of staying on the safe side.
So the size of the fibers, how do you choose the size of the fibers? Because using some data on how long opposing fibers are, thickness of opposing fibers. So it's experimental.
Yeah, based on experimental data.
This corresponds to huge bundles.
Sorry?
This corresponds to very large bundles.
Yes, exactly. Yeah, exactly. Yeah. So and of course we have now static crosslinkers. We just initiate the network, drop in a lot of crosslinkers, and then we let the model run. We also have static photon regions. But I'm having some new data where we're changing that, and I'm also having some new data where we changed that I don't have here, but where we change the network. Yeah, we can of course change fiber density. And by the way, this is so the cell will stay like this. So this is where you see that indeed there's force exerted on the cellular plots model. So the cell tries to retract, it will then lead to prohibitive large energy change, such that it cannot retract further. But this cell really wants to disappear, but it cannot because it's held back by the fibers. Well, yeah, we can compare this with all kinds of data. For example, this was data from work by the Dell and Mac groups, where they had cells in an extracellular matrix that is trying to contract. these are fibrous in this case. So, and they observed, for example, that the fibrous is accumulating around the cells. And what we did is, in our model, we time average locations of the fibrous, so you get a picture that looks very similar to the microscopic image. And indeed, what you see, if you may now have less cross linkers, that indeed also the cells accumulating this matrix around it, and that is kind of understandable because if you don't have any connections to the surrounding fibers, then you can just pull the fibers towards them, and the rest will stay. So part of this effect is of due to and not fully percolated now. Then we can also do things like, which is also in this experimental work, when you put the cell and you take the cell out. Well, if you have a kind of loosely connected network, you put the cell and it contracts, and you take it out, and the network won't recover. If you have a really stiff, highly percolated network, so it contracts, you take the cell out, and the thing will bounce back. Oh yeah.
Now I checked, 10 minutes.
Okay, perfect. And indeed, what you see is that it recovers completely, but if you have a little less percolation, then it's intermediary stage where it recovers a little. But we don't have true viscoelastic, so it's kind of viscoelastic behavior, meaning due to sliding of some of the fibers. We don't have full viscoelastic behavior because for that we need to have dynamic binding and unbinding, which is working well. And then we can also do, yeah.
Situation of the cell shrink. What's the experimental condition? It's an apoptotic cell?
No, that's a good question because what did they do? And because I have to go back to the paper because I had the same question. So I remember at one point I had the answer, but how? No, you ask me again. I have to go back to the paper. Yeah, but they did something convincing. I was convinced when I read it, but that's again what they do. But it's not a product, but they have some kind of experimental way to kind of kill the cell in a way that it's also not affecting the rest of the matrix, because that's of course really what you want to do there. Yeah, this is just an example of other things you can do. So this is published a VAM experiment with a compacting cell, and then you see the direction where it's pulling the matrix strength stiffness a little bit. We can also do that in our simulation. So we have the contractile cell. You can see that in some places it leads to bundling of the collagen bundles. And at those places, you see that the matrix becomes stiffer. And so what you then can do is analyze this further. So there are a couple of sectors where you see that some places there's the matrix is just less dense. So if you keep on poking there, there's no real strain stiffening. So that once you select out these places like those sectors where there is still strain stiffening, you see that it nicely decays and substitutes in these kind of systems. The outlook. So this is now work by Numerical, the PhD student in my group, and he's looking at how he can bring back those dynamic focal lesions into the system. And the nice thing of having this molecular dynamics framework is that we can now replace the focal lesion springs. So instead of having this part directly connected to the cellular side, he puts a little spring in between, a very stiff one, and the extension of this spring will tell you how much tension there is in this place. So that we can feed again to the same type of equations, and also we can have dynamic formation of these focal lesions, as you can see here. It's one fiber in cell. I really like watching this movie because it really shows how the forces are interacting and how the It should be the third one, I think. Anyway. Yeah, there it was. Yeah. All right. So yeah, he has this additional spring now. And he can, at least he can now model cell spreading using that. We're not yet at cell elongation. But at least now, as we saw in the initial model, the cell becomes bigger, it's making it stiffer. So you can measure that. These are 3 simulations. It was one where the big gap was made in the labels. So that's the outlier there. Of course, we can play with the geometry of the extracellular matrix. In this way, with this case, we have stretching ECMs. And yeah, I think I could stop here. I would like to briefly, I will just briefly show some movies, some slides on how we can also model the intracellular dynamics. This is work with Luca Gioni, who's in Leiden University, in the physics department, where we now introduce liquid crystal model inside the cells to model anisotropic contraction, because so far we had only isotropic contraction. I can just show you. So this is some test cases. These was theory by Luca Gioni and where they showed that indeed if you have anisotropic contraction, usually the young plus model predicts circular arcs between completion points, and now you can show these should be ellipses. And indeed also can, if we put this in the cell model, you get also ellipses. So actually there are also discrepancies in these, and actually the reviewer asked, we're still working on this review for months, reviews for months. Reviewer asked a really good question. Simply, why don't you put, instead of the, these were hand fitted, why don't you put the theoretical curves in there? And that was a really good question. It turns out that it reduces this discrepancy. So I'm really happy with that question. So let me see. Yeah. So of course the next step is then to put this kind of detail again in this cell model. So we also have anisotropic contraction and also feedback between cell shape and isotropy of the cell of the cytoskeleton. Yeah, and of course, I had way too much material. This is actually about collective migration, evolution of collective migration, but I think I'm really out of time.
Yeah.
So if you want to know more about this, have a look at the paper or ask me about it later. And of course, we want to go to multicellular systems, so we can do that already with more simple models. And where we had we kind of put a lot of detail on computational dynamics into the Hamiltonian terms and the work terms. And with that, we can also explain how cells respond to static stress. But they have to pull a little bit themselves in order to make that happen. So with that, oh, I hope none of my students are watching, because they said, please replace this this group picture, because I'm closing my eyes, I'm looking in the wrong way, and so I said, oh, you know, I changed the picture, and I did in another version of this presentation, but I've got to do it in this one. So I'm so sorry if anybody of you is watching. So sorry, but thank you so much for doing all this wonderful work. Thank you, funders, and thank you for your attention.